Hi, this is lecture two for microbial metabolism and the adaptation microbiome by Diane Marie Brache Smith. That's me. And in this part of the lecture, we're going to get into metabolism, ATP, and redox, and why that's important to understand these types of things, at least on the basic level, when it comes to creating growth media for different types of bacteria that you're trying to isolate.
Metabolism is the sum of all chemical transformations in a cell. There are two categories of chemical transformations. There's catabolism, right? I like to think of the prefix kata. Think of like catastrophe. That means to break down. So kata, to break down. And what catabolism is doing is it's breaking down a molecule into smaller parts. And each time it breaks down a molecule, these bonds release energy. Catabolism is an oxidative process that loses electrons to release energy, and then this energy can be used to perform anabolism. And for anabolism, I think of the prefix ana. Um, Anabolic steroid users are building muscle, right? Ana is to build, so they're building proteins when they're using steroids. Um, and anabolism is a reductive process that uses energy from catabolism, and what it does is it breaks these bonds. It remakes these bonds, right? And making those bonds takes energy, and which is a way that you can store energy or that you can build macromolecules such as lipids, polysaccharides, proteins, nucleic acids, anything that a living earthly needs to live and thrive. So jumping into the cell itself, so these processes of catabolism and anabolism. The nutrients for biosynthesis are used in analysis and anabolism, and these to create macromolecules for the cell. Um, Catabolism, which uses light as an energy source or chemicals as an energy source, will to break bonds, and this energy release will be used for motility and transport of cell nutrients. It can be used as energy in anabolism for biosynthesis to create cell components, and in the process, you're going to have waste products, and these waste products include alcohols, carbon dioxide, and any reduced electron acceptors. Cells can conserve energy in the form of ATP by coupling its synthesis to release energy via oxidation reduction reactions, also known as redox reactions. ATP is a phosphorylated nucleotide, right? You see ATP here, right? And it is the main energy currency for all cells, right? So these phosphate groups will be broken apart to release energy, or they can be put together to create energy. Now, I'm sorry, to store energy. But when you create this bond, you are going to need energy to repair that bond. So ATP breaks down to ADP. ADP is then phosphorylated into ATP, and this basically manages all the energy in a cell in manageable quantities, which means a lot less is wasted. In oxidation reduction reactions, where electrons are passed in the form of an electron donor to an acceptor, electrons must be coupled with atoms or molecules. Every redox reaction consists of two half reactions, where one substance donates electrons and thus becomes oxidized product, while other substances accepts the electrons and thus becomes the reduced product. And I know you guys are probably like, oh my God, this is overwhelming. I'm going to show you how this kind of translates to understanding how energy is exchanged in all metabolism, so you can kind of sort of pick out what nutrients you're going to put in to a nutrient growth medium to grow the kind of microbes with the metabolisms that you desire. Here you can see redox of carbon organic compounds that are generated by photosynthesis broken down by respiration. Um, these are in oxic conditions here, and these are the ones that are more energetically favorable. And for each of these processes, there are genes that create enzymes that catalyze these processes, right? And these genes are sort of the ones that I start to look at when after you've isolated a microbe and you're going into the bioinformatics. It's like, well, are the genes there to carry on these processes that I believe that there's evidence to be occurring in the system. We can use redox pair potential to predict what microbes are in an environment and what pairs they use for energy by using the electron tower. The substances at the top of the tower are strong reducing agents, and they readily donate electrons, and those at the bottom are strong oxidizing agents. They readily accept electrons. Um, as you can see here, CO2 glucose redox pair, it gives you 24 electrons, right? That's a lot of bang for your buck, where maybe like a sulfate hydrogen sulfide pair only gives you 8 electrons. So if a microbe is capable of doing sulfate metabolism and carbon dioxide glucose metabolism, which one is it going to go for if it has both available? It's probably going to go for this one. Now that same organism, let's say, doesn't have any glucose in its nutrient medium, but it does have sulfate. It can grow and use sulfate as an electron donor. Aerobic conditions provide the most energy for respiration, and bacteria use electron acceptors that provide the most energy and move down the tower as they're depleted. For example, the aerobic oxidation of glucose can produce close to 3,000 kilojoules per mole of glucose. That's a lot of energy, right? Now, while sulfate reduction can produce close to 500 kilojoules a mole, which is significantly less than what it can with glucose metabolism. You can see the difference on how energy sources can grow a diverse array of microbes. Now we'll go over carbon, nitrogen, and sulfur metabolism in depth as it relates to the Aptasia microbiome.